I'm Dr. Don Milton. I am a physician scientist here at the University of Maryland School of Public Health and the Maryland Institute for Applied Environmental Health. So when we think about flu, we usually think of seasonal flu that comes every winter, sometime in the winter, or early, late, it can vary. And that is with viruses that are already what we call human adapted. They have figured out how to infect us, and once we become immune to them, they don't seem to affect us again. And it can go several years without much influenza activity and then have a really bad year. And the reason for that is that over a period of years, the virus changes, and once it's changed enough, it becomes able to infect us again. Our antibodies and other immune defenses don't work against it so well anymore, and so then we catch it again. But every now and then the virus makes a major change, and the virus has the ability to swap genes with other viruses, and when it does that, it can become very different, and all of a sudden we have no immunity to it, or almost no immunity to it, and then it transmits very rapidly. And that's what happened in 2009. The virus was what's called a triple reassortment. It had swapped genes with 3 different versions of viruses coming from birds and pigs and humans. And that triple reassortment created a virus that very few people under age 50 had any immunity to. And so it spread like wildfire. Summer camps in Maine, and have kids coming down with flu, it was really unusual circumstances. That's what happens when you have a pandemic. We were really lucky. That virus turned out not to be very lethal. It was not a highly virulent virus. We didn't know that at first because the first reports out of Mexico suggested that it was a pretty bad virus. But once it spread more, it turned out not to be so bad. It killed people, but the total death toll was nothing like we could have seen it had it been a highly lethal virus. And then you have the thing about flu, why it's very hard to get rid of it, is because most of the flu viruses aren't actually human viruses. They're bird viruses, pig viruses, and they're always circulating out there in the wild bird populations. And they become a big problem for farmers raising chickens because they get into the chicken flocks and they can wipe out millions of chickens. But that is a big reservoir of these flu viruses, and what we worry about is them jumping from the birds into people. And that's when we talk about the avian viruses. And this avian H5N1 is a highly lethal avian virus. And that is the threat, if that jumps into people and does it in a way that retains its virulence, we will have a big problem on our hands. Interestingly, with this new experiments, there were 2 groups. One group created a highly transmissible virus related to the avian virus in such a way, by combining it with the 2009 pandemic virus, that ended up making it more like the 2009 pandemic virus. It lost its virulence. It didn't kill the ferrets. The other group mutated it in a way, without combining it to an existing human virus, that made it transmissible, but it retained its virulence, and it was highly lethal to the ferrets. That is a really dangerous virus. And we can see that there are a couple of ways this thing could get into the human population. We can only hope that if it does the former and not the latter, and that we get a mild virus out of it. But we can't count on that. We need to know what's possible and what we can do to prevent the spread of it. We're working right now on an NIH-funded study that is looking at whether putting surgical masks on people who have flu will reduce their shedding of virus into the air to a great enough extent that it can control influenza. So can we limit the spread of flu by, when we're sick, wearing surgical masks? We expect soon to start working with funding from CDC to extend this study beyond just characterizing people who have flu and how much they're spreading and whether surgical masks will work, to actually comparing people who have acquired the infection naturally out in the community and however that happens. And that's part of the issue, is we don't know exactly how that happens. We know that it has something to do with coughing and breathing, but we don't know whether the droplets that infect you are the very big ones or are real little tiny ones that float around in the air. And to answer that question, after we characterize people here in Maryland and in BC with naturally acquired infection, we're going to go over to Nottingham in the UK, where they're going to be artificially infecting people with flu, and putting them into a small room in a hotel with people who are susceptible to the flu. And some of those susceptibles will be wearing face shields and some won't. And we'll see whether the people wearing the face shields and the ones without the face shields are equally likely to get the flu. If that's true, then it's the tiny particles that float around and can go around the face shields that are causing it. If only the people who don't have the face shields on get infected, then we know that it's the large droplet spray when you cough and you sing and speak that are spreading the flu. And the reason that we're going to be doing this study on the people in Nottingham is we don't know for certain that when we artificially infect somebody with nose drops with flu in it, that they get flu the same way that somebody naturally catches flu, and that the kind of infection and the way they shed virus into the air is the same. So if the artificially infected people don't shed virus the same way naturally infected people do, we won't be able to use that experiment to define how it spreads in the real world. So it's critical that we know that those people in Nottingham look like real flu cases. And that's what we're going to do. This machine here is called the Gesundheit 2, or the Gesundheit machine, or G2 for short. And what it is a device where a person with influenza or another respiratory infection can come into this little booth that we've built here, where it's nice and warm and humid inside, and sit on a high chair in there with their face into the cone, and the cone will collect all of the breath that they exhale and bring it down through a device here that will collect the different particles in different size ranges and allow us to see whether they have virus being spread into the environment in their exhaled breath.